lecture, we are going to continue our discussion about experimental design and all of the nuances related to experimental design. Specifically, we're going to talk in this section about categorical versus continuous design. And that will lead us into a discussion of something called an analysis of variance, or an ANOVA for short, which is a very powerful type of statistical tool that will become very important to you as we continue on through this class. A quick recap of what we talked about in the last video. Replication is the inclusion of multiple experimental units, whatever your unit of replication, whatever your treatment units are, representing a single treatment in the same experiment. Replication is important for finding patterns. Of course, one is not a pattern. Coping with mistakes that might surface where you have to throw out one or more replicates. Balancing confounding factors, which we didn't really talk about that much. And finding differences if the effect size is small, and recall that the effect size is the expected difference between averages in your experiment. Measurements that are close in space or time to each other are often non-independent. Recall the birdhouse example where birds are attracted to yellow birdhouses or red birdhouses. I can't remember, but because of the proximity of the one to the other, birds are attracted to the other non-desirable birdhouse. Dependence among replicates can be reduced by randomizing treatments, and the randomization of methodology and of treatment location are both important. I will be looking for randomization in your experimental design. As far as randomization goes, randomization is when you have no systematic way of assigning order to anything. There is no underlying systematic bias towards how we assign things. Whereas haphazard is what most of us always use when we say we're assigning randomness. It is when there's an underlying systematic way. You are a bad random number generator. If I asked you to choose a number 1 to 4, 1 to 5, you would almost always choose 2 or 3. So let's kick this discussion off with an amusing video, The Drunken Monkeys of St. Kitts Island.
This is the sleepy island of St. Kitts in the Caribbean. "300 years ago, vervet monkeys were brought here from West Africa along with slaves serving the rum industry. Escaped monkeys acquired a taste for alcohol by eating fermented sugarcane left in the fields. Today, they satisfy their thirst by raiding local bars. They have learned to be sneaky. Picking the right moment is everything. For years, the monkeys have been studied for insights into our own drinking habits. Just as we vary in our taste for alcohol, so do the monkeys. Some do anything for an alcoholic cocktail. But just as some people are teetotal, so are some monkeys. These reject alcohol in favor of soft drink. Significantly, the percentage of teetotal monkeys matches the non-drinkers in the human population. In line with human habits, most drink in moderation. 12% are steady drinkers, and 5% drink to the last drop. This similarity between us shows that a liking for alcohol is determined mainly by our genes. After each daily raid, other human parallels soon appear. But unlike us, monkeys that are heavy drinkers make better leaders, respected by other monkeys. They seem to tolerate leaders that monkey around. Like monkeys, our taste for alcohol began when we scoured the forest for ripe fermenting fruit. Food and alcohol became linked with intoxicating effect.
Based on this video, I hypothesize that older primates prefer stronger alcohol. All right. Now, I have no idea. This is a very interesting study. I have no idea if this is actually a hypothesis. I'm just using this as an amusing example. There are psychologists and evolutionary biologists that are studying the vervet monkeys to understand our own evolutionary association with alcohol. I don't know if this is actually a hypothesis, but we're going to go with it. Now, I predict that older vervet monkeys on the island of St. Kitts will prefer drinks of higher alcohol content in choice tests. That is a much more specific prediction. If we go back to our hypotheses, you can see that it's very general, very broad, but it is directional. And here's my prediction that older monkeys on the islands of St. Kitts will prefer drinks of higher alcohol content in choice tests. Now, here's where we start creating an experimental design. We've given a hypothesis statement. We make a prediction statement. The next thing that we will write in our papers is, to test this hypothesis, I will do something. I hypothesize this. Given my hypothesis, I will do this. And to test my hypothesis, I will ... In an experiment, the independent variable is the measurement that predicts something. I predict that older monkeys, in this case, age is the predictor. It is the thing that is going to influence our response. The dependent variable is the response. Age is our predictor, and age will predict what alcohol content they will prefer. So alcohol content is our response or our dependent variable. It depends on the independent variable, which is age. The dependent variable is not necessarily caused by the independent variable. Remember that we can never truly determine causality. Correlation does not equal causality. But we say that the independent variable might cause the dependent to change. It at least influences the dependent to change. Independent and dependent variables can be either categorical or they can be continuous in an experiment. A categorical variable is classified, are variables that are classified into one or more unique categories. For example, if we were to look at age of vervet monkeys, we could number them 1, 2, 3, 4, 5, etc. Or we could lump them into categories such as infant, toddler, adolescent, reproductive adult, and post-reproductive adult. These are categories that have a meaning in our experiment and have meaning in terms of the life cycle of the vervet monkey. Our response could also be a categorical variable. For example, we could say light beers, porters, stouts, wine, whiskey, rum, etc. These could be the categories of what we would choose as our responses. We could record whether they choose light beer or wine, etc. The choice of categories strongly depends on your hypothesis. We hypothesized about alcohol content. If we hypothesized about alcohol content, we probably should not use the categories of wine versus rum because wine and rum have very different levels of alcohol content, but they also have very different levels of color, sweetness, and flavor. And we're not hypothesizing about color, sweetness, and flavor. We are hypothesizing strictly about alcohol content. Hence, those categories are not good for our hypothesis. There are other confounding factors that differ among a porter versus a stout versus a rum versus tequila, etc. There's a lot of other things that the monkeys might or might not find desirable. Tequila. Your categories should differentiate only the factor of interest. It is extremely important, and I will call you out in your experimental design for this. A lot of people have difficulty in eliminating confounding factors. Please pay attention to this when you're compiling your experimental design. Hence, a better option in this experiment might be to choose one of these alcohol types and simply add more grain alcohol to increase the alcohol content. In other words, let's take wine, which has 15%, and simply, we know that they like wine. We know that they like wine. We've done the observations. Let's add more alcohol to wine, but we're not going to change anything else. We're just going to add alcohol to increase the alcohol content. We're not going to do Cabernet versus Champagne. We're not going to do white versus red. We're going to stick to the exact same color, the exact same flavor. We're just going to change alcohol content. That way, we stick to alcohol. We do not change anything else. So, to emphasize, whether your predictor or your response is continuous or categorical really depends on the nature of your response. Let's go through a couple examples. What is the effect of different fish species on the number of snails in a lake? In this case, our treatment, which is going to be fish species, is categorical. Maybe we look at the effects of walleye versus the effects of perch and the effects of trout. The response is the number of snails that we are going to measure following the addition of one of these fish. So we have a continuous response and a categorical predictor. On the other hand, perhaps I hypothesize about the effects of fish mouth size, not fish species. Now my predictor is continuous. But you might be saying to yourself, well, wait a second, isn't that introducing the confounding factor of now these are different fish species? And you are correct, absolutely. Maybe they have different appetites. It would be better if we could surgically enhance or de-enhance the mouth size of trout and look to see if that mouth size alters the number of snails. But sometimes we just can't do that. Maybe we look for trout that have larger or smaller mouths but are the same length, and we could do that and do it that way. I don't know if that's possible. In lieu of that not being possible, we are going to just select different fish that have approximately the same appetite level but have different mouth sizes, and then look at the number of snails. Another example. I'm going to come back to this pesticide example time and time again because this is just what I study, and so it's or what I have studied, and so it's at the forefront of my brain. What is the effect of pesticides on lakes? I should also mention that if you're planning on studying environmental science in the state of New Jersey, learning the effects of chemical contaminants in aquatic systems is actually really big business. Now, if I'm concerned with the effects of chemicals with different target organisms, for example, if I'm concerned about pesticides that affect insects, also known as insecticides, or pesticides that affect plants, also known as herbicides, then my predictor is categorical. I'm looking at insecticides versus herbicides versus fungicides versus, um, uh, um, I think I've ran out of sides. There might be one more. Anyways, if I'm concerned with the dose of the chemical or the reactivity of the chemical or the size of the chemical, the size of the molecular size of the chemical, then my treatment is going to be continuous. In your experimental design, I will be asking you to tell me whether your predictor and whether your independent variables are continuous or categorical.
Eyes out of my way. You make the sunshine brighter than Doris Day. Turn the bright spark into a flame. My beats per minute never been the same. 'Cause you're my lady, I'm your fool. It makes me crazy when you act so cruel. Come on, baby, let's not fight. We'll go dancing, everything will be alright. Wake me.
Some categorical variables are actually what we would call ordinal. Now, I'm sure most of you have seen this at a carnival where you slam a hammer down onto this thing and the bell goes up. All right. And depending upon where it goes to, they tell you whether or not you're weak sauce or whether you're super strong. Strength can be measured as a continuous response. For example, we would measure bicep dimension, and that should relate to strength. However, this device in a carnival doesn't do that. It ranks your strength according to the height of the slide, the bell going up, or so yeah, the slide going up to the bell. So it ranks your strength as 5, 6, 7, 8, 9, 10, or 11. And you get put into those categories. Now, you might be tempted to say that these are continuous, except continuous variables actually need to be able to take any value between 5 and 6, like 5.5 or 5.4, and that's not the way that these devices categorize strength. You are either a 5, a 6, a 7, an 8, a 9, a 10, or an 11. That's it. You can't be anything between those. So this might be a continuous, but because they can't take any value, it's actually a categorical variable that we call ordinal because the categories are meaningfully ordered. All right. Indeed, these numbers represent categories of strength, but they're ordered. Ordinal variables will often arise when you would like a measurement to be continuous, but whatever device you're using makes it unable to measure that measurement on a continuous scale. Let me give you a real life example. When I was in the tropics, I wanted to measure the effects of a chemical on plants, and I didn't have the ability to measure photosynthetic rate. I just wanted to look at how green they were. So I said, suppose I hypothesized that plants are greener when they are watered more. Well, I also did not have the ability to measure how green the plants were on a continuous scale. I would need to take a picture of them, put them into a computer, and then use the computer to tell me how green they were on a continuous scale of, I Photoshop stuff. So I don't have the ability. All right. I have, in this experiment, I had 5 treatments of different watering regimes, but I really couldn't take pictures of them and I couldn't put them into a computer. I couldn't rip them out of the ground and take them back to the lab with me. I had to make a snap decision about how green each one of these leaves were. And so I made a rank. I had my assistants rank the leaves in order of brown to green, where 1 was brown, 2 was yellow brown, 3 was yellow, 4 was yellow green, and 5 was green. And I asked them to simply provide a rank for me. Now, this is a continuous-ish variable, except it could not take any measurement between 1 and 2, or between 2 and 3, or between 3 and 4, or between 4 and 5. It can only take 1, 2, 3, 4, or 5, those integers. That's it. So this is actually a categorical variable that is ordinal. The type of variable that you have in your experiment determines the type of analysis that you will conduct. You really need to memorize this table. This table is everything. This table is your cheat sheet to understanding what you will need to do with any given experiment. If your independent and dependent variables are continuous, then your design is a regression design. Let me show you what I mean. If my predictor is monkey age, all right, now monkey age is weird because monkey age is usually 1, 2, 3, 4, 5, and those are discrete integers, but we could actually measure age as 2.34 or 4.10 or 4.1 or something like that. We could actually measure age on a completely continuous scale. And my response is alcohol content, which is a percentage, which is continuous. This is a regression. And the way that we view regressions is with an XY plot, where the predictor, which is now monkey age, goes on the x-axis, and the response, which is alcohol content or the predicted, dependent variable, goes on the y-axis. This is what is known as a regression design. Now, something to consider with a regression design, I have to measure monkeys of a wide range of ages in this regression design. Otherwise, I don't have any idea whether my hypothesis can be supported. For example, if I chose to measure monkey ages that were only, let's say, in this range right here, in the black range, then I would not actually be able to detect whether there was indeed an effect of monkey age on alcohol content, because it doesn't look as though there is in that age range. However, if I also measure young and old, the blue dots on the left-hand graph, then I do see that they are, that alcohol content is related to monkey age. In addition, you really want to make sure that your predictor distribution, your independent variable, is approximately uniform within the sampled range. You don't want to be measuring all young monkeys and have just one older monkey. And that one older monkey turns out to be an outlier because, well, actually, you don't know if it's an outlier or not. You have no idea. If you only measured the black monkeys in this, the young monkeys, you have no idea if alcohol content is actually related to monkey age. But if you did measure that old monkey, but you only measured one old monkey, then you really still have no idea. You only have that one old monkey. You have no replication in that upper age range. Now, if the independent variable is categorical, whereas the response variable is continuous, this design is known as an analysis of variance, or an ANOVA. Now, I am not going to cover the other two options here in any detail, at least not right now. Maybe we will try to get to it towards the end of the semester, because honestly, regression and ANOVA are the two most important parts. So not only am I telling you just to remember this simple table, I'm also telling you that you really only have to remember the first row of this table. An analysis of variance occurs when your dependent response is continuous, but your predictor, your independent variable, is categorical. Let's go through an example. I suspect that eating different types of Chinese takeout food before an exam has different effects on a student's grade. I don't know about you, I love Chinese food, and the greasier, the better. I love American Chinese food. I unfortunately have not been to China, so I don't have any experience with actual Chinese food. I just love the sugary sweetness of and the oiliness of the American stuff that we can get. And so I hypothesize that consumption of more oily food is positively correlated with performance. Notice how this is a broad hypothesis. It does not talk about the type of food. It does not talk about the specifics of the food. It just talks about oily. That's my broad hypothesis. And I could technically test this with any type of food, but I'm going to test it with Chinese food. A true test of this hypothesis would be to feed oil straight to students. Forget about the General Tso's versus the broccoli and chicken. Forget about all of that. A true test would be to feed oil to students. Now, this seems unethical, so we're not going to do that. Instead, I am just going to choose a variety of meals that vary in oil type. They will vary in other things like nutrition, but it's the best that we can do. Now, recall that this is a similar problem that we ran into with feeding vervet monkeys different types of alcohol. We don't want to vary the sweetness or the color of the alcohol. We just wanted to vary the alcohol content. It's more feasible to vary the alcohol content and use that as a predictor. It's a little less feasible and probably way more unethical to just feed humans different levels of oil. In this experiment, my predictor is food type. I only have one predictor in this experiment, which is food type. Within that predictor of food type, I present students with 3 different meals: General Tso's, egg foo young, and beef and broccoli. I say that I have 3 treatments of my predictor. Or I could also say I have 3 levels of my predictor: General Tso's, beef and broccoli, and egg foo young. This type of design, where you have a single predictor, a single independent variable, is known as a single factor design. Please, please note that the independent variable is different from the treatment. The treatment, or treatments usually, is how many different levels of your predictor there are. In the monkey case, how many different age groups are we looking at? How many different, what's the age range we're looking at? That's the variation within my independent variable, but I generally only have one independent variable. However, the power of an analysis of variance design actually lies in something called multi-factor designs. For example, let's suppose that I hypothesize students will do better on exams if they eat a fortune cookie with their meal. All right, pretty good. Let's say that fortunes just raise their spirits and they're more likely to do better on an exam if I give them a fortune cookie with their meal. So I provide them with 3 different types of food, 3 levels or 3 treatments of my first predictor. But now I have a second predictor, which is the presence or absence of a fortune cookie. Now I thought about this experiment and I thought maybe I could do a control where I just look at whether it's the cookie and not the fortune that enables them. But then I realized that this is honestly probably the most depressing thing that I could ever think of happening to me in my own personal existence, that a cookie without a fortune happens. So I'm just going to get rid of that. I don't even want to think about it. For the sake of simplicity, let's ditch that and let's just look at whether or not the meal type influences their exam score and whether or not the fortune cookie influences their exam score. So now notice there are two factors or two predictors, or in other words, two independent variables, type of meal and fortune cookie. There are 3 levels of type of meal and 2 levels of fortune cookie. Okay? In our experiment, we give students General Tso's chicken. And we have 10 replicates in which we give students General Tso's chicken with fortune cookies and 10 replicates where we give them without fortune cookies. Beef and broccoli, we have 10 replicates where we give them with fortune cookies and 10 replicates where we don't have fortune cookies. And by the way, replicates in this case are students, and we're going to measure their exam scores. And again, we have egg foo young, 10 replicates with fortune cookies and 10 replicates without fortune cookies. So in other words, we have 30 individuals that ate fortune cookies, right? So you have 10, 10, and 10. And you have 30 individuals that did not eat fortune cookies. Again, 10, 10, and 10. You can count them up. We have 20 individuals that ate General Tso's, and we have 20 individuals that ate beef and broccoli, and we have 20 individuals that ate egg foo young. In total, we have 60 total individuals that we're looking at, or 60 total replicates. First, we explore the effect of food type on exam score. Notice this is slightly different from our single factor, or also known as a one-way analysis. In this case, each bar includes people that ate and did not eat fortune cookies. It's only different in that we are including all levels of the other factor in this. Each one of these bars is the inclusion of all replicates, regardless of whether they did or they did not eat a fortune cookie. There doesn't seem to be a difference. It seems as though people who ate General Tso's chicken had the same performance as students that had beef and broccoli or egg foo young. Now, we are going to take a look at the other independent factor, without fortune cookie and with a cookie. And we can see that the students that ate fortune cookies, regardless of what meal they ate, we're not confusing ourselves by considering meal within fortune cookie yet. We're just looking at the 30 replicates of individuals that ate fortune cookies and the 30 replicates of individuals that did not eat fortune cookies. The individuals that ate a cookie seem to do much better. All right. Regardless of what their main meal was, we can conclusively say, I mean, I know there's no error bars here, whatever, we can conclusively say that cookies improved exam score, or at least cookies were related to an improved exam score. All right. What we've just done, looking at the effect of meal type regardless of cookie, and the effect of cookie regardless of meal type, we have just examined what are called the two main effects, or the two main factors of the experiment. But what if the effect of a student's meal depended on whether you ate a fortune cookie?
The time has come for this cookie. Nom 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 nom. Oh, nom 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 nom. Ah, bye bye.
And herein lies the incredibly powerful use of an ANOVA to detect interactions in nature. What if students that ate a fortune cookie generally did well but they did mostly well when they ate General Tso's. You wouldn't really be able to tell that from this graph, but if we divided this graph up into all of those respective combinations, students that ate General Tso's without a fortune cookie, students that ate General Tso's with a fortune cookie, students that ate beef and broccoli without a fortune cookie, students that ate beef and broccoli with a cookie, students that ate egg foo young without a cookie, and students that ate egg foo young with a cookie. We can see from this hypothetical silly example that without a cookie, all of the students did the same. With a cookie, they generally, on average, did perform better. However, they seem to perform most well when given a cookie and General Tso's chicken. They also seem to improve when given beef and broccoli with a cookie. Egg foo young, I guess it's just gross. I personally don't like egg foo young. That's why I made this the example. All right. We will talk far more about these interactive designs in an experiment. It is extremely powerful and an extremely useful way of doing statistical analyses. The take-home point is that ANOVA designs are powerful ways of testing independent and interactive effects of multiple predictor variables, multiple independent variables.
The time has come for this cookie. Nom 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 nom. Oh, nom 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 nom. Ah. Bye bye.